Hello, Professor. My name is Daniel, and I'm going to be presenting on amyotrophic lateral sclerosis. So what is this disease? Well, it is a multi-system neurodegenerative disorder characterized by motor neuron degeneration and progressive muscle weakness. It was originally considered a motor neuron disorder purely, but it was later reclassified as a neurodegenerative disorder due to its extramotor characteristics. It is a disease with high heterogeneity in that over 20 genes are linked as causal factors in ALS. It is difficult to treat, not only because of its multifaceted uh, causal structure, but also because of the mechanisms involved. And there are no available cures or treatments, no effective treatments, I should say, at the moment. And that the prognosis is quite poor, in that patients who are diagnosed with ALS often die after 3 years of symptom onset. So it is very difficult to treat. The most we can really do for them, for patients with ALS, is provide a multidisciplinary approach that involves respiratory aid and nutritional aid, things of that nature. And I should also mention that the reason for death in most instances is respiratory failure because of muscle weakness in those respiratory muscles. So in terms of clinical presentation, there are two primary considerations when diagnosing ALS. One is upper motor neuron and lower motor neuron dysfunction in one region of the body. Another one is lower motor neuron or LMN dysfunction in two regions. And this has to be accompanied by progressive muscle weakness. And the muscle weakness tends to begin in distal limb muscles. This is something called limb onset ALS, as opposed to bulbar onset ALS, which I will discuss somewhat more in depth later. But essentially, that's ALS that begins with muscle weakness in the face and the head and the neck. Extramotor manifestations are relatively common, in particular, cognitive and behavioral changes. And this occurs in about half of ALS patients. And there is a correlation with frontotemporal dementia as well, with 10 to 15% of patients also having FTD. And the incidence case is around 2 per 100,000 persons per year, so relatively low. In terms of etiology, causal factors, it's a combination of genetic, environmental, and aging-related factors, but it is mainly genetic. Over 20 genes have been linked to ALS, and these are some of the most common ones. It is a highly heritable disease. Most of the variants in the phenotypes of ALS are due to genetic factors. And generally, they are distinguished. ALS is distinguished as sporadic or familial. That is, do you have a family history of ALS or do you not? Sporadic. And these are some of the risk factors. Certain genotypes, protein repeats, ATXN2, old age, male sex. Males tend to develop ALS at a higher rate than women, but only by a slight margin, and environmental exposure. In terms of pathogenesis, there are three main theories of ALS pathogenesis. The dying forward hypothesis essentially argues that ALS has its origins in the cortical spinal neurons, and degeneration makes its way from there through monosynaptic connections between the cortical spinal neurons, the upper motor neurons, and the alpha motor lower neurons. That's the dying forward hypothesis. The dying back hypothesis, in contrast, argues that pathogenesis begins in the muscle or in the neuromuscular junction of the lower motor neuron with the muscle, and retrograde transport of toxic materials is what induces the systemic-wide ALS phenotype. The independent degeneration hypothesis essentially argues that both can be true. Pathogenesis can begin in the upper motor neurons or the lower motor neurons, or both. And pathological mechanisms will occur along the motor neurons through their connections, but that neither one is necessarily more dominant than the other. And there are variable patterns of origin progression in this disease. And depending on the origin, where it starts, how it starts, the mechanisms at play, there can be a very different progression. As we mentioned before, only some patients have cognitive changes in the brain that are pathological in nature. Others don't. Why this occurs is not exactly for certain, but it could be because of the origin, the pathogenesis of ALS, where it begins and where it progresses from there. Some important pathological mechanisms. These are some of the important pathological mechanisms we can see here. I will detail most of these. Some of them I'll skip or mention briefly, just for the sake of time, and just so our discussion remains mostly relevant to muscle physiology. So, one of the first things we can talk about is cytoplasmic aggregation. This is mainly due to RNA metabolism, dysregulation, and it is a primary neuropathological signature. In fact, cytoplasmic aggregation of TDP43, a DNA binding protein, is present in over 95% of ALS cases, which makes it quite prominent, obviously. So essentially, mislocalization, accumulation, aggregation of proteins reduces buffers against oxidative stress, which leads to several pathological mechanisms. We'll get to oxidative stress in a moment, but essentially how this works is proteins, RNA binding proteins mainly, and some DNA binding proteins that are mainly localized in the nucleus. They are present in the cytoplasm, but mainly localized in the nucleus. That's where their function is. For a variety of reasons, are localized to the cytoplasm, where they aggregate and disrupt a variety of different mechanisms, such as proteostasis. And this exacerbates certain neurodegenerative pathways, which we'll get to shortly. Another key pathological mechanism is glutamate excitotoxicity, where there is an imbalance between excitation and inhibition in the synapse, and this leads to hyperexcitability and disrupts cell function. This is mainly due to reduced GABA levels. GABA is the inhibitory neurotransmitter, where glutamate is the main excitatory neurotransmitter. So by having reduced GABA levels, this imbalance occurs, essentially. And it might be worth it to go briefly over how this mechanism works. Normally, we do have a picture here. So essentially, glutamate is released by the presynaptic neuron, in the upper motor neuron, I should say, into the synapse. It binds to AMPA receptors mainly, and this initiates a postsynaptic response, which is typically in the form of a graded potential that, if strong enough, will trigger an action potential. Now, in the figure here, we can see that only 20% of the glutamate actually make it to the postsynaptic neuron, and around 80% are recollected, recycled by astrocytes. They're converted to glutamine, etc. So there's this upregulation of glutamate, but reduced GABA levels, and as Osherson mentioned, reduced glutamate transporters among astrocytes. Then we have this upregulation of a synaptic response. This increases calcium influx, and a variety of different pathological mechanisms are impacted. So here is a better figure demonstrating this. We have the normal physiological mechanism here and the pathological mechanism here. We can see the upregulation of the glutamate, the dysregulated astrocyte, where these transporters are not working effectively, and a variety of different pathological mechanisms ensue. Take particular note of the reactive oxygen species, which is upregulated. We can discuss that in a minute. But essentially, yeah, the postsynaptic neuron succumbs to excitotoxicity, and ALS pathogenesis or pathology occurs as a result. So related to that, and one of the main hallmarks of ALS is oxidative stress. ROS production impairs a number of important regulatory mechanisms, such as protein synthesis and mitochondrial function. It is linked to post-translational modification of RNA-binding proteins. This results in cytoplasmic localization and aggregation. It also impairs communication and interaction between the mitochondria and the endoplasmic reticulum. It disrupts several key mechanisms of cell regulation and induces RNA dysmetabolism. So we've discussed our reactive oxygen species in class, but essentially it impairs a number of different processes and is also interlinked with many different pathological mechanisms. And we'll discuss more later, but it is related to this more overarching positive feedback loop and cycle that kind of exacerbates the development of ALS. But in terms of mitochondrial function, ROS can induce this, mainly in the form of electron transport chain dysfunction, which then leads to higher reactive oxygen species production, reduced ATP production, and calcium homeostasis. And this is, again, part of this positive feedback loop, which induces further oxidative stress. So we have these reactive oxygen species that interact with the mitochondria and cause disruption, which then further causes ROS production through the electron transport chain and its interaction with oxygen. And yeah, we have this positive feedback. And glutamate excitotoxicity is also relevant here because it disrupts calcium homeostasis. It increases a calcium influx. The mitochondria responds by increasing calcium entry, and then this further disrupts mitochondrial dysfunction, which we see this loop that kind of begins to manifest. Another important mechanism here is the impairment of proteostasis, which results in ER stress, which then results in cell death. So proteostasis is the process of balancing protein generation, newly translated proteins, and degradation of misfolded proteins. And this is thrown off balance by ALS pathology. Protein accumulation and imbalance damage the endoplasmic reticulum. This results in an ER stress response, which induces various signaling pathways, resulting in an unfolded protein response to restore proteostasis. And this causes upregulation of chaperones and cellular clearance systems as a means of reorganizing the system and trying to put it back in balance. But if the ER stress persists, then the signaling pathway will shift and induce apoptosis, which means neurodegeneration. And ER stress-induced autophagy can increase cytotoxicity. We also have the impairment of the ubiquitin proteasome system. The ubiquitin proteasome system is essentially a way of recycling proteins. Ubiquitin tags misfolded proteins and takes them to the proteasome, which then degrades them. And similarly, the autophagy lysosome system is a way of clearing out damaged proteins. So these are also impaired, resulting in further disruption of proteostasis, further ER stress, which leads to further cell problems in the cell and eventually cell death. Neuroinflammation is also a key process here. It occurs due to protein accumulation, mitochondrial dysfunction, ROS production, essentially a variety of different pathological mechanisms we've already mentioned. But what it essentially does is activates microglia. These are macrophage-like glial cells, which are they serve as immune surveillance cells in the central nervous system. When they are activated, they release inflammatory factors, which activate astrocytes, which then induces a variety of cytotoxic developments in the astrocyte. One of those being impaired glutamate transportation, also upregulation of reactive oxygen species in the astrocyte, and this will eventually lead to cell death. An interesting tangent of this is that early neuroinflammatory responses are actually neuroprotective. This is mainly through a transcription factor known as NF-κ beta, which again is it helps in this process. It results in microglia cell proliferation and lymphocyte infiltration, which is protective of motor neurons. That being said, it only seems to be this case in early stages of neuroinflammatory responses. In later stages, we see this activation of the astrocytes, these reactive astrocytes, which was seen in an earlier slide, and a variety of pathological mechanisms ensue. Oligodendrocytes, which are also glial cells, are the glial cells that produce myelin sheaths that insulate the nerve. They also degenerate through a variety of pathological mechanisms. I keep on repeating that word. I'm sorry about that. But yeah, it's another key process that is quite important. Here we see, actually, a good visual of this. We have the quiescent astrocytes, which through to a variety of different genetic mutations or inflammatory factors released by the microglia induce a transformation into reactive astrocytes, which results in ROS, upregulation, ER stress, proteostasis disruption, pretty much everything we mentioned, ion imbalances, yeah, and eventually apoptosis. Neurodegeneration is the result. So, okay. So, axonal defects. Obviously, the ability of the axon to transport cargo is extremely important. I mean, that's how signals are propagated. That's how calcium influx becomes meaningful. That's how it releases neurotransmitters. Well, this system is impaired because of these different pathological mechanisms, because of reactive oxygen species and so on and so forth. It's a number of different mutations we can identify that impair the neuronal cytoskeleton. One is prolifin 1 and tubulin alpha 4A. This disrupts the micro tubule network. We also have point mutations in genes encoding dynactin 1, which disrupt the dynactin complex. This disrupts the dynein motor, which prevents stabilization of cargo transport. Likewise, mutations of kinesin 1 impair anterograde transport of cargoes along the microtubules, which again prevents synaptic vesicles and other cargo from transporting their materials to the terminal axon so that neurotransmitters can be released and proper cell communication between the cell body and the axon terminus can be achieved. So yeah, we come back to this theme of positive feedback. Every one of these different pathological mechanisms, while independent of one another in some ways, are also deeply interlinked and deeply causally rooted, if you will. At the center stage here, we see oxidative stress, which we discussed, and this induces mitochondrial dysfunction, leads to ROS production, RNA dysmetabolism, leads to protein aggregation, ER stress. These are all interlinked. One causes the other, and that recauses the initial causing factor. And in the end, we have neurodegeneration, cell death, and impaired axonal transport. It may be worth it to just go through it one more time. I mean, we have the glutamate excitotoxicity, where calcium influx is upregulated. So, and this is only one pathway, keep in mind. There's other pathways that can achieve the same result, but calcium influx is upregulated. This leads to higher calcium entry within the mitochondria. This disrupts the electron transport chain, which increases reactive oxygen species production. As a result, there's an increase in RNA dysmetabolism, which leads to protein mislocalization and aggregation, which leads to an ER stress response. It tries to counteract this through different mechanisms, the unfolded protein response pathways. But eventually, if persistent enough, even this mechanism fails and cell death is inevitable, and exonal transport, the cytoskeletal structures that regulate that system will completely fail. The neuron is unable to generate a meaningful postsynaptic response, and synaptic vesicles can't transport their materials to the terminal axon. So the lower motor neuron has very little means, assuming it's not dead yet, the cell body isn't dead yet, it has very little means of actually activating the muscle, activating contraction. So we have muscle weakness. And if pervasive enough, at a point, it does become pervasive enough in that the respiratory muscles fail and the person is unable to breathe without support, without external ventilation, and that and the patient is essentially unable to sustain their life. So it's a very vicious cycle with its a variety of quirks. So we do also have regional discrepancies within ALS. I mentioned before limb onset versus bulbar onset. Essentially, limb onset is when the pathogenesis begins in, or the symptoms begin in the limbs, where we have muscle weakness that becomes progressively worse in the arms, in the hands, or in the legs. Bulbar onset, on the other hand, begins with weak muscle control over muscles in the face and in the neck. So in particular, speaking and swallowing. And this is considered much more severe and maybe related to other more deeply pathological mechanisms. We also interestingly see that fast twitch fatigable motor neurons are the most vulnerable. In this case, we're talking about three classes of motor neurons: fast twitch fatigable, fast twitch fatigue resistant, and slow twitch fatigue resistant, which are of course related to type 2X, type 2A, and type 1 respectively. And this makes sense. If we just take a moment to think about what this means, the fast twitch fatigable motor neurons, which are associated with type 2X, have much less mitochondria and have higher metabolic loads. So they're much more reliant on the energy systems at play. And because when mitochondrial dysfunction ensues, they're much more vulnerable because they have no other means of effectively producing energy. They don't have backup reserves of mitochondria that can pick up the slack, if you will. So they are the first ones to go. They're the most vulnerable in this case. So in terms of future prospects, unfortunately, no cure or effective treatments are available right now. Only two drugs are currently approved, riluzole and edaravone, but these aren't particularly effective and they only extend life by a few months on average. Now, stem cell research is offering promising solutions in the future, and they have produced a few drugs that are currently in clinical trials. For example, ropinirole is a dopamine receptor agonist, which combats a variety of different ALS phenotypes, and in particular prevents cytoplasmic aggregation of certain DNA binding proteins, RNA binding proteins like DDP43, which, as we mentioned before, was a prominent neuropathological signature in most ALS patients. Also redigabine and bocetinib. Digabine, I believe. I'm having trouble. I can't remember what redigabine does or bocetinib, but these were also combative towards some of the key pathological mechanisms of ALS. With that said, in conclusion, ALS is a highly heterogeneous disorder. It's extremely complex, multifaceted in its mechanisms and its causal factors. Over 20 genes contributing towards it. Each mutation affecting different pathways within the body. And again, this discrepancy between cortical spinal neurons and degeneration and lower motor neuron degeneration, how pathogenesis in one can trigger a completely different progression of ALS than the other one. In fact, this may be the way we can explain why in some patients we see cognitive and behavioral changes, and in other patients we don't. It could be because of where exactly the motor neuron lesions occur. If they're more uh, geared towards the brain, if they're upper motor neuron, then perhaps that results in cognitive and behavioral changes. If they're lower, then perhaps that results in worse motor features, so on and so forth. Again, it's very difficult to treat because of this multifaceted nature, but there are promising treatment solutions being explored. So that is my presentation. These are my references, and yeah, thank you so much.